The 658th Assembly of New Asia College of CUHK in 2022 to 2023 starts now. This assembly is jointly organized by Yan Kwok Yung Lecturer in Life Sciences of New Asia College, and we are so honored to have Professor Tsai Ming Daw, Distinguished Visiting Chair, Institute of Biological Chemistry, Academia Sinica Taiwan, to share with us on life science research, important developments in the past 50 years, and major challenges ahead. We are also pleased to have Professor Hector Chen, our college head, to introduce the speaker. Professor Chen, please.
Professor Tsai, honorable guests, fellow alumni, colleagues, and students, good evening. I'm here to welcome you to join this 7th lecture of the Renguorong Lecture in Life Sciences. New Asia College and the School of Life Sciences have co-organized this lecture since 2015 as a result of generous donations to New Asia College from the family of Dr. Ren Guorong, founding chairman of the Department of Biology of the college. Each year, we have invited different distinguished scholars, biologists, and life scientists to come to our college to share with us their views on certain aspects of life sciences. The lecture series have broadened the knowledge of life science among students, staff, and the general public. Today is the first lecture of the lecture series. It is my great honor and pleasure to introduce Professor Tsai Ming Daw to you. Professor Tsai is currently the Distinguished Resident Chair, Institute of Biological Chemistry, Academic Sinica of Taiwan. And he is also the professor at the Institute of Biochemical Sciences, National Taiwan University. Professor Tsai received his bachelor degree in chemistry in 1972 from National University of Taiwan and PhD in medicinal chemistry from Purdue University in 1978 from the laboratory of Professor Hans Voss. He was a faculty member in the Department of Chemistry and Biochemistry at the Ohio State University from 1981 to 2006. Subsequently, he moved to the Institute of Biological Chemistry of Academic Sinica Taiwan and served as the director during 2006 to 2014. He was elected to Fellow, American Association for the Advancement of Sciences. He's also an academician of the Academic Sinica and Fellow of the World Academy of Science. Professor Tsai's research subjects in the past 40 years has been in the interface between chemistry and biology. His main interest is the mechanism of specificities of enzymatic reactions and protein functions. He probes these problems by applying emerging methodologies in structural biology, including nuclei magnetic resonance, X-ray crystallography, and recently cryo-EM and X-ray free electron laser. Professor Tsai has made significant contributions in the mechanism and structures of phospholipases, DNA polymerases, kinases, acarine repeat proteins, and the FDA domain proteins. Many of these proteins are relevant to DNA damage, DNA repair, and cancer signaling, as described in nearly 300 research papers. Apart from being a scientist and researcher, Professor Tsai has trained over 60 PhD students and over 40 postdoctoral fellows, and many of them have become highly successful scientists. He is also actively involved in developing infrastructures and services. During his tenure at The Ohio State University, he has established a chemistry biology training program and served as the director for 8 years. He also served as the director of the campus Chemical Instrument Center for 14 years, overseeing the development and operation of NMR and mass spectrometry. In academic Sinica, he led the Taiwan Protein Project to establish the academic Sinica cryo-EM facilities and to develop a programs to use X-ray free electron laser to determine reaction intermediates of DNA proteolysis at ultra-fast timed scales. Without further ado, may I now invite Professor Tsai Ming Daw to give the lectures on life science research, important developments in the past 50 years and major challenges ahead. Professor Tsai, please.
Thank you, Professor Chan, for the very generous introduction. And I'm also very happy to see so many young students and also see a few my scientific and personal friends here. Particularly, I know Professor Henry Chan for a long time, Henry Wong for a long time. Good to see him today, and also Professor Rap Chu, and of course, Director Kimball Wong. So it is my great honor to deliver this hopefully useful lecture. Certainly it is a very important lectureship. The reason I choose to talk about this subject is because we have all been through the pandemic, and I think all of you should all agree that life science research is important. Some of you may not know that most of the technologies that have been used to fight the COVID-19 have been developed, discovered only in the last 50 years, or even the last 20 years, or even the last 10 years. If we had not been doing extensive life science research in the last 50 years, we would probably not have done as well against COVID. So let's go to the basics. We all consider what is our basic needs in our life. And food and health certainly are the basics, and that's life science. Then we need infrastructures and communications, and that's physical sciences. And as you will see from my talk, the life science is actually integrated or very dependent on physical sciences. Many of the important discoveries in life sciences was due to the development in physical science. And certainly, we also need entertainment and arts and music, that's humanities. So no matter what you do, it's all right. You could choose to focus on one, but you could do all three aspects. So in my own case, I was a chemistry major, that's the physical science in college, but I've been doing life science research in the past many years. And in my college, I spent a lot of my time actually doing music, playing in an orchestra and quartet, string quartet. And this picture was taken in 1971. But many of the young students here, most of you young students here were not born yet. Even your parents probably were not born yet. So to the young people, last 50 years seems nowhere. Okay? But to some of us, I mean myself plus maybe a couple of friends there, 50 years is not a long time, and we have we have witnessed what has happened in 50 years. Okay? It's hard to imagine there was no internet 50 years ago. Okay? No computers. Well, there was computer, but no laptop computer. So let's go to the basics. And this is what you would learn from high school, I'm sure. So the central dogma, DNA keeps the information through RNA, and then proteins do the function. So information is important, but the functions, the molecules that do the work are even more important. Okay? So to simplify the system, I mean, there are so many different things we could talk about, but you could summarize the subject, the main subject, into actually two aspects. One aspect is the level of molecules. And at this level, we are talking about genes or nucleic acids, okay? And then we're talking about proteins and enzymes, and then drugs or vaccines, okay? So these are all molecules. And then at the level of organisms, we talk about biology, medicine, agriculture. We talk about pathogens, human diseases, and food and nutrition, and that include agriculture. Yeah. So if you simplify it this way, then it's not too complicated. And for my lecture, I'm going to focus on actually two major areas. One is the genes and nucleic acids, and then proteins and enzymes. And then in terms of the other drugs and vaccines and diseases, I'm going to use COVID-19 as an example to go through some of the issues. So let's go through the main subject here, the second aspect. That is, I'm going to go through the development related to genes in the past, and that could be summarized by 6 Nobel Prizes. So the first one is the solution of DNA structure. Okay? And that was Nobel Prize in 1962. So this is what we know now, Watson Crick base pairs. Okay? And of course, we know how important DNA is. Okay? It determines everything. Okay? And then the DNA needs to be sequenced. Now we could sequence DNA very, very fast. You know, in the COVID period, you know, you could sequence anything quickly. But 50 years ago, that wasn't that easy. So the Nobel Prize in chemistry in 1980 was awarded to DNA sequencing and recombinant DNA. Recombinant DNA means you could take a piece of DNA from one organism and play around with it and or put it into another organism and express the protein in a different organism and so on. You could do many things. Okay, so those people got the Nobel Prize, and that was only 1980. And then 1993, that's not too long ago, okay? But, you know, you heard about the 3 letters PCR almost every day, right? Every minute in the last 2 or 3 years. And that was discovered only 20, 30 years ago. And that technique is the technique that could amplify the DNA, trace amount of DNA, so that you could detect what, you know, whether you got infected by the virus and so on. And then site-directed immunogenesis is a technique that allows you to make a specific mutation on a specific site of DNA so that the protein that is expressed by the DNA would change the amino acid at a specific position and would have a different function. So you could manipulate DNA this way. And one of the major achievement by humans was the completion of Human Genome Project in 2003. Actually, only 92% was completed at that time, and the rest, the remaining 8% was completed only last year. Okay. But at that time, I remember in the cover of Time magazine, or every magazine journal shows about this Human Genome Project, and there was a prediction that in our generation, we could live, because of the completion of this project, we could live to 120 years old. Okay? But I don't see that happening yet. Maybe you have a chance. So, well, let's see what happens next. And then the next one, most recent, is the prize is given to a technique called CRISPR. Okay, this is a genetic scissors-like technique that allows you to edit almost anything, DNA from anything. Okay, and this could have a huge impact. It's already having huge impact. And last year, the Nobel Prize was given to the sequencing of ancient DNA that allows us to analyze the evolutionary history of humans. Okay. So you look at these Nobel Prizes and you can see, wow, that's so great, and that seems to be so remote from us, from you. But actually, these are all done by normal people. Okay? Normal students could get a Nobel Prize someday. Okay, one of you, one of us. Okay? So in the future, this is only my humble opinion. I don't know enough. I could not predict enough. I could not even predict what happened in the next 10 years. But I think the speed, accuracy, and price of DNA sequencing will continue to improve. Personalized medicine will become more and more routine. That is, we could analyze a patient's genetic information and then do the prescription, do the treatment based on that, okay? And then the genetic editing technique will be further used to develop cell-based therapeutics and many more. So then let's move to proteins and enzymes. Okay. So the atomic resolution of protein or amino acid or atoms is in the scale of angstroms. An angstrom is 1/10 of a nanometer. Okay. Now, it is important to understand or to know, to have the resolution, the atomic resolution of molecules, because it allows you to know the specificity. See, the living systems, the most important word for the living system to me is specificity, the function. Every biological function has some kind of specificity, and that specificity is achieved by the arrangement, spatial arrangement of atoms, okay, that make up amino acids and proteins and so on. So specificity is always also related to the chemistry, chemical basis of specificity or chemical basis of protein function. And that kind of information then allow us to design inhibitors or drugs to attack or to solve problems or to attack certain targets. Or then the development of protein drugs such as antibodies. So many Nobel Prizes have been awarded to the development of methods to solve protein structures, as well as the solution of protein structures. There are three major methods for determining the atomic resolution of protein structures. One is X-ray crystallography. And let's go through that, a few Nobel Prizes for that. So by the name, this technique uses crystals. Okay? And only in 1962 that the first protein structure was solved. That's the structure of hemoglobin. And this technique is based on the diffraction of X-ray by the atoms in the crystal. And based on the diffraction pattern, the structure could be deduced or solved. And this started with a laboratory scale apparatus instrument. But nowadays, X-ray crystallography is always performed in synchrotron facilities. So here there are two rings here. A small ring is the old one, and a big ring is a new one. In Taiwan, the big ring is called Taiwan Photon Source. That was built only less than 10 years ago. And this circle, I mean, some of the many arrows comes out. That means this synchrotron radiation could be used in many different purposes. Okay, they could, they could be built beamline stations at different places and do different things. And this is inside the facility. And this facility is open to the world, and I know some of the colleagues from this university from Hong Kong have come to experiment there also. Now, in terms of the proteins that was discovered, DNA polymerase, so in 1959, Nobel Prize was awarded for the discovery of DNA polymerase by Arthur Kornberg. And then the structure was solved later in 1980s by Tom Stites. So this is an enzyme that replicates DNA. Okay, DNA information needs to be replicated, and that is done by a protein. And this protein is called DNA polymerase. Yeah. And then in our cells or the particles within cells, these membranes and molecules need to go in and out of cells, so they go through membranes. So membranes have channels. So even water have channels. Okay, so water channel, potassium ion channel, there are many, many different channels. Yeah. So if you, at that time, only 20 years ago, if you solve the structure of one of these channels, you could get a Nobel Prize. You got a chance. And in 2006, the discovery and solution of, the solution of the structure of RNA polymerase by Roger Kornberg. And then the ribosome is the protein complex, the particle called ribosome. It's a place where the protein synthesis occurs. And you got a Nobel Prize also. And more recently, in 2012, the discovery of G protein coupled receptors called GPCR it's a type of receptor. We have many receptors in our cell membranes also, which do signaling. It receives signaling. Okay, there are many signals. Okay. Or occur receptors, and they receive extracellular signal and induce intracellular functions. So that's about X-ray, and there's another technique called NMR, nuclear magnetic resonance, and that is based on magnetic field. If you put a protein molecule in a magnetic field, then every atom in the protein molecule will experience somewhat different, slightly different magnetic field. So they will all have slightly different resonance signals. And if you could assign each of them and determine their relationship, their between space relationship, then you could solve the structure, atomic structure. And 2 Nobel Prize was given to NMR. One first for the development of the methods, and then in 2002 for the application of this method to solve protein structures. And at this time also the mass spectrometry also received the Nobel Prize. Mass spectrometry is also another very, very important, powerful technique. And all of you know about MRI, okay, magnetic resonance imaging. That is based on the same principle as NMR, but they got a separate Nobel Prize for the contribution to medicine. Now, the most recent discovery, breakthrough in this field, in structural biology, is cryo-EM, okay, cryo-electron microscopy. So this technology existed maybe for 20, 30 years, but in 2015, 16, there was a breakthrough in the resolution. That is, it has been improved to the level that you could look at atoms. Okay. Now, cryo-EM is a technique that you could actually see the molecule. Okay. And I'll explain to you how do you see the molecule. Okay. But it does not rely on magnetic field. It does not rely on diffraction. Okay. So this picture is taken from the Nobel Prize homepage. So it's very simple. So suppose this is a microscope. So this is individual protein molecules. So each protein molecule may consist of many hundreds of atoms. But if you spread the protein molecules, you just dilute it to a single molecule. You know, they don't overlap. Each of them has this, their own space. And then you shine the beam, shoot the beam through, then you will get an image like a shadow. Okay? So the image will be different for every molecule because every molecule is randomly oriented. Okay, they are all random. Okay? So in principle, you have a different shape from all different angles. So if you use computer, could put them together, the computer will be able to give you a program software to give you the real shape of the molecule, real details. And resolution is a problem. No. One image is not enough, so you could what you do is you do many, many images, thousands of images, or hundreds of thousands of images, and then you search for the same images and overlay them, put them together to enhance the sensitivity. That's all. So in other words, the microscope actually look at the shape of the molecule. Okay, not by your eye directly, your eye through the microscope. So we also took advantage of this technique as quickly as we can, and established the Cryo-EM Center in Academia Sinica in 2018. And by 2019, we have this nice picture of a protein molecule. You could, enjoy the picture for a minute or so. So this is a 12-mer, dodecamer. So this complex has 12 protomers or subunits. And it is a hexamer of dimer. So you could see this is a dimer. So we will zoom in, and then you will see that protein binds ligands or binds metal ions. So this is 2 magnesium ions, and this is a substrate or inhibitor, or this is NADH, a cofactor. Now this technique, Cryo-EM, does not require crystallization, so it has advantages. Now, give you some idea about the scale. What is the atomic scale? We talk about nano all the time, right? And so since this is a relatively large complex, the diameter is about 5 nanometer. Okay. How small is it? So we have a microscope here. So the biggest object that I know that has been observed, but not by eye directly, also by telescope, is a black hole. Okay. The difference between black hole and a protein molecule, I put 2 things here. One is our Earth, and the other is the apple. The diameter of the apple is 5 centimeter, and this is 5 nanometer. The difference is about 10 to the 7th. 10 to the 7th. But because this is diameter, for the volume, we need to make a power of 3 again. So this is the difference between an apple and a molecule. If you could imagine how large it is. But you could imagine the difference between an apple and our Earth. Okay. That is 10 to the 8th, and then the power of 3. And then you could try to imagine the difference between a black hole and the Earth. That is 10 to the 9th. Okay. Now, there are many black holes in the universe. Okay. And there are a lot of space between black holes. So it's hard to imagine. So there are so many things you could study, you could learn. It doesn't matter what you do, as long as you do you make an effort, do your best. So about the future, again, I couldn't predict too much about the future. But most proteins function as complexes. They don't function as individual molecules. They function together with other proteins. So we need to have more structures of multi-protein complexes and bound with ligands. And then ideally, we like to have all of these structures at their functional state. That means when these molecules are in cells, not in instruments. Yeah. And or even in humans, in tissues. And that is a technique called cryo-electron tomography that has been developed, has already made good progress. Excuse me. And then structures are only snapshots. Okay? The function is a dynamic process. So we need to know the functional process of the structure. We need to have a time dependence of structures. So we need to see structural changes at real time. And that, I will talk about this subject tomorrow in the School of Life Sciences. And I'm sure many of you are curious in your mind that what can AI do? There are already many programs for structural prediction by AI, and there will be more in the future. But I think this is something we don't have to worry about. All we have to worry about, the AI is based on, at least in the prediction, structure prediction, it is based on the information we provide, okay, based on the structures that we have already solved, and then AI get the information from there. Okay. But what we do as scientists, researchers, what we do is to do the unknowns, okay? And then all of what we do need to be tested experimentally. So AI could help us in some way to do preliminary screening work and so on, particularly in drug screening, but it should not replace the scientific work. It may help us with our work in the future. And then drug and vaccines, I will just mention briefly because there is so many of them. So this is 1974 vaccine, chickenpox and HIV protease inhibitors, MRI in 2000. More recently, cancer immunotherapy is a very good field, important field. There's a Nobel Prize also. And then I'm going to talk about COVID-19 next. And for the future, I think the messenger RNA vaccines will be further developed for other diseases, such as cancer, stem cell research, cell-based therapeutics. Okay, so let's spend a few minutes on the COVID-19. So I think most of you should have seen something like, pictures like this. So this is the viral particle that have proteins on the surface, and then the most common protein is the spike protein. Spike protein will find a receptor in the host, and that receptor is ACE2. And by this interaction, the virus will infect human cells. So this molecule inside here, this is the RNA. So this is a genomic RNA. So this sequence was available as early as the January 10th of 2020. Okay, you know how early, how soon that is. Now with genomic sequence, we could deduce a protein sequence. With protein sequence, we could solve protein structure. From protein structure could help us to examine the infection mechanism and drug development. And all of these have happened very, very fast. And many of the work were done in China, in Hong Kong, everywhere in the world. So imagine how soon this was. Okay, this was February of 2020. The structure of COVID-19 spike protein was published by Science magazine. And it is a structure of trimer. So there are 3 protomers, but the 3 protomers are not identical. So this is one of them, this is the other of them. They are slightly different at this region. Now this region is called RBD, receptor binding domain. this is the domain that intrudes into the ACE2 receptor. So that's an important region. So this is called RBD up and RBD down. So that means this protemer is more likely to infect or have a better infection ability than this protemer. So when later different variants of the virus have been evolved, then all the scientists will rush in to look at the structure of their spike protein to see how the conformation have changed, and that could help them develop therapeutics such as antibodies or vaccines. And then again in March, only 1 month later, the complex of the RBD and ACE2, the receptor protein, have been reported. And this came from laboratories from Beijing and Shanghai. And with that, these researchers were able to compare the interaction between SARS-CoV-2, that's our COVID-19, and the SARS 20 years ago, to look at how the interaction with the receptor differs. Okay, now this gets into more details. We will not get into those details today, but the point is you could do a lot of comparisons. So here you could compare the Omicron with the Delta, for example, and there are some differences in the interface between the RBD and the receptor. And this work actually also came from Chinese labs. I think one of the Hong Kong labs is here. I think Chao Su is from one of the universities in Hong Kong. And then a lot of antibodies have been developed, and the antibody binds to the RBD in order to block the spike protein from binding to the receptor, ACE. That's how antibody drug works. Okay. And structures have been solved also. The first drug that's on the market was remdesivir. Yeah. And that has been, look at also the structure of remdesivir bound with RNA-dependent RNA polymerase. That's another, the replication protein. Okay, that's it. The final few minutes, it's not about science, but my suggestion of the future or for the young people, that is, what are possible problems ahead of us? We don't know if the pandemic will come again or different ones will come again. Yeah. So we have to prepare to fight it. And then the global warming would really be a problem. So some places in the world will have too much water, other places will be too dry. That will be our problem. Okay. AI could be very beneficial, but it could be very dangerous also. Okay, like anything else, like nuclear power, yeah, it could be very beneficial, but also very dangerous. We don't know how AI will be used. In your lifetime or next 50 years, I think chemistry and biology and medicine will be merged. I was chatting with our director, Chen. He's a medical doctor. He said he'd like to see molecules in patients, and that will be possible in the next 20 years, 30 years. Infectious diseases would always be around. Humans will live longer and better if we don't destroy our planet. The impact of AI could be huge and beyond imagination. considering that 50 years ago there was no internet and no cell phones and no laptop. Not until I was in college. So if you are interested in a research path for your career, college education is still basic. You should consider, you should get a basic, well-rounded basic education. You have a major for sure. Yeah. But overall, it's more basic. And doing an undergraduate research is very useful. Yeah. Constantly foster your problem solving and self-learning abilities. Because eventually you have to self-learn. You cannot be taught by anything, everything. Think positively, like your Xiaoge. I like your Xiaoge. Yeah. Do what you like and like what you do. Okay? And some personal experience, finally. Science always follows a good logic. That's in my view. There's no magic. Combining good tools and good thinking, logical thinking, yeah, we could solve problems. Depth and breadth are equally important. Okay, there seems to be contrasting, but they are equally important. Science is a field where you could make friends with your competitors. That's very important. Very, 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 very nice. Scientific mentorship or friendship often lasts forever. Like what I have with many of the colleagues here. We have met in scientific meetings and we become friends and friends. For example, this is Professor Chen in the middle. She is my undergraduate research mentor. And every year, his former students, we still get together and celebrate her birthday. She's about 90 years old now. And in the center is my PhD mentor, Professor Heinz Frost. So he came to my sort of retirement party. It's not a retirement, but I have served over 25 years in Ohio State before I moved to Taiwan. So they gave me a retirement party. And I have another career here in Taiwan. He just passed away a few months ago, unfortunately, and we're having a ceremony, memorial. So the rest of the people are my former students at Ohio State. They came to my this activity. And these are some of my former students who are doing teaching in universities, around the world in different places. So this is, other than doing science, this is most the rewarding thing for a professor is to have your former students or your mentors know each other and have this relationship. And this was taken just before the pandemic, at the beginning of it. And this was when I was overseeing two important operations. One is the Taiwan Protein Project. The other is just the newly established Academia Sinica Korean Center. And with this, I'd like to close and thank you for your attention. Thank you.
Let us again express our gratitude to Professor Tsai for his wonderful lecture. We will now move on to the Q&A session of the Assembly. May we invite Professor Hector Chen to moderate the Q&A session. Professor Chen, please.
So thank you so much for your very brilliant talk, very inspiring. One obvious thing is you have gone through a very successful career, publishing a lot of papers, and then build up your team, and then advance the fields substantially. But now that perhaps I start the first question while I'm waiting for the question online or from the floors. Did you come across any failure in your pursuit of knowledge?
Well, it's a good question, but I try not to use the word failure. I say something doesn't go through. The biggest failure we could have is when a paper is rejected by a journal or when a grant is rejected by a funding agency. And when that happens, I try to recover overnight. I try to be sad for half of a day.
So you went to bed?
Yeah.
And then forget the things?
Forget about it next day. Yeah.
And then come back again this morning?
Yeah.
I
I think you need to think positively. I think many things have two negative and positive aspects.
So now is the time for questions. Is there any questions from the floor? If there's a question, please come to the middle. There's a microphone there. Could you please voice out your name and department and then start the questions? There's a student there.
My name is Tim, Timothy Isa. I'm from the business department. I'm just curious if you think it's feasible at all within the next 20 or 50 years that diabetes, type 1 diabetes could be cured. Also, thank you for your talk, and it was really interesting.
Sorry, could you repeat the questions?
I was just curious if you think that type 1 diabetes could be cured.
Type 1 diabetes, could it be cured?
Do you think it's feasible?
I think it could be, yes. I think certainly. I think anything could happen in the future. Yeah, but when, we don't know. And they could speak in Mandarin.
Yes, actually we could entertain questions in Mandarin, if you like. Any more questions from the students, from our colleagues down there? Perhaps let me ask another question.
Sure.
All your research based largely on the availability of all these very fantastic equipments. But it's very difficult for one to write a grant to apply for this equipment. So how will you develop a team or a lab by purchasing all these very fantastic setups?
Yes, this is a very good question. There's different levels to answer this question. When I was a student, of course, I would think to get new equipment is a job of the department chair or the director or the deans, not the job of the student. But when I was a student, and at that time, NMR instrument was becoming available. And when there's a new instrument in the college, so right now cryo-EM is becoming available. So what a student could do is you should compete for that accessibility. So for example, I was always willing to go to the lab at night in order to have access to the instrument. So that's one thing you could do. Now, when I become professor, then I'm more willing to take on administrative positions to help people. So I would volunteer to write grants for funding of new instrument, because a new instrument could help myself, but it could also help other people.
Right, yeah.
So I think the key thing is you think about yourself first. That's very important. Don't think you don't have to think about yourself. But then you think about other people also. Something good for you will be good for other people also. And then you could convince other people, oh, this is good for all of us. Then let's get together to see the director and ask for funding.
Not me, yeah. Yes, maybe. Yes, please. There's a student ask the question first, and then followed by KP.
Professor, thank you for your amazing talk. And as we know, you do life science. So may I ask, what is your philosophy of life?
Your philosophy of life.
你生命的哲学。
生命的哲学，这个问题很大啊。
这个好像
那从你这个人生的经历，你有什么精神可言？就你整体的那个精神，你有没有什么感悟呢？
我想很简单的讲就是努力工作嘛，然后努力思考，做你喜欢做的事情，然后不要做坏事情，然后我最希望的就是时光能够倒流，但是现在是不可能的事情。我最希望我是像你们一样，现在还坐在那里，
那么年轻，
那么年轻。那可是我也能够回想我当初像你们一样的时候，我在想什么，我在做什么。那我觉得人生能够经过看过，人生能够这样走下来，就是非常难得、非常完美就是非常非常珍贵的事情没有说什么太好太不好所以这个就是我觉得，那也不要想 win everything， 你不会 win everything。
Okay, thank you.
Then KP, please.
Hi, Ming Daw. Since you have trained a lot of tens and tens of students, PhD, postdoc from your lab, I'd like to hear, and many of them are actually very successful in their own research career. I'd like to listen to any tips and experience that you can share with me. I'm also a mentor myself.
Okay, thank you. I think I show two pictures. One is my undergraduate mentor. One is my graduate mentor. And I think I was influenced by them. And both of them are very kind people, very open and free and encouraging. So I see there are all kinds of different mentors, different style, and some of them, they could all be successful. But my style is to give them more freedom and encouraging and don't watch over their shoulder all the time. But there are some professors who do that, and they are very successful also. It's like parents and children. Not all model will fit all. And even the same parent, I got 3 kids, and they are all very different. And so your student, they are very different. And actually, you need to treat every student differently. When I was younger, I was able to have more interaction with each student, and sort of some students need more guidance than others.
Okay, thank you, Ming Daw.
Another question from my colleagues. Jimmy?
Professor Tsai, as I told you, I'm an engineer. Thank you again for your lecture. Now, at the beginning of your talk, you told us that there are basic needs of life. And then you also show us a picture of you playing violin. Now, if you go back to the time when you are 20 again, would you choose the same career path, or you'd rather be a musician?
Well, musician is not certainly not for me, but some of my siblings are musicians. But in terms of the, I have three siblings, and my parents used to say, I like to study because I don't play well. My brother and sister, they are professional musicians. And I couldn't play like them, so I just focus on study. So anyway, the point is, it doesn't matter. And when at that time in Taiwan, I ended up in chemistry. That was not my choice either. I was more interested in physics. But it doesn't matter because I think I like to do. I can like anything I do. So when I do something, I try to do it well. And now I think chemistry is the best major to have as an undergraduate, because chemistry allows you to get into almost any other field. I mean, after graduate from chemistry, you could go to medicine, you could go to engineer. You could go to life sciences, or you could go back to physics. So that's my philosophy.
Thank you very much. I think you remind me of what the great tennis player, Roger Federer, what he said. He said he could be a great table tennis player. He could be a great football player as well, as long as you enjoy what you're doing. Thank you very much.
Yeah. There's a question from the online platform. The question is, could you please give more idea about the AI applications in protein structure research and drug design? What do you think about the future potential and challenge of AI in drug and atomic level research in life science? This looks like very specific questions.
Well, AI is around for at least 10 years. I mean, it has been used in protein research. There are different programs. See, the issue of AI is it is accelerating. And since it is accelerating, we don't know how soon, rapidly it will be. But I think the problem of AI now, the accelerating aspect of it is more in the routine area application. Okay. See, whatever AI does, it has to be, if you are talking about accuracy, then there's a limitation. You can ask AI about specific questions, scientific questions, paper that I published. AI couldn't answer it right. I tested it. Yeah. But to write a routine data is very useful. So AI is useful in screening. Okay, so in drug screening, for example, what we need is to from a drug library of 1 million compounds to narrow down to 2,000 compounds. I think AI could do that. But from 2,000 compounds down to 200 compounds, to 20 compounds, then we need to do experiments.
Yeah.
So it saves us a lot of labor in the first screening. Yeah.
Okay. So is there any questions from the floor? Yes? This gentleman, could you go to the middle of the hall at the back and use the microphone there?
Hello, good evening, professors. I'm a final year student in Bachelor of Pharmacy.
Pharmacy
yes.
final year students.
And actually, I have a degree in biomedical sciences. And I'm currently wondering, should I go to the research pathway or to the clinical pathway? Because, you know, life has so many choices, like you have said, you can choose either to be a musician or to be a scientist. So I'm quite curious, what makes you to decide that you want to be a scientist instead of a musician? And what is the basis for you to make that decision? Thank you.
Okay, thank you. This is an important question. Other question is,
maybe I can repeat the question. The question is, how would Professor Tsai decide at the time when he have to make a decision of whether going to be a musician or whether to be a scientist? I think this student is facing similar problem because
clinical.
he have to choose a clinical path or a scientific path.
Well, I think you talk to people around you, your professors, advisors. I mean, they know you better, they could help you, but they should not make decisions for you. You should follow your heart and make your decision yourself, but you should talk to people. And there are many decisions you have to make in the process of your life, but don't be afraid to make decisions. And once you make a decision, don't look back. That's all I could say. Every decision is a good decision. You cannot predict whether it's better go this way, or it's better go that way. No one could predict for you. Okay, but you get the information, you think, and you listen to your heart, and you make a decision, and then go with it. That's it. There's no failure. No, just as long as you do what you like, and you like what you do.
Okay, thank you.
Okay. Any questions? If there's no more question, perhaps it's time to stop the lectures today. So let us thank you, Professor Tsai.
Once again, thank you, Professor Tsai, and thank you, Professor Chen. May we invite Professor Chen to give souvenir to Professor Tsai and have a photo taking on the stage. Thank you. Professor Chen